All we're doing this week, today we're going to spend time talking about retroviral drugs. Wednesday I kind of have set aside as like review day. I thought it might take us longer to go through HIV than it has. So as of right now, what we'll do is we'll finish up anything we need to from today. And then I think the rest of the time we can use for you guys to just be, I can come with some review problems for us to be working on for prep with the exam if you'd like. I'll probably just take directly from either old exams or from what's already available up on Blackboard for review questions to work through. If you have multiple choice questions you want to submit for the last exam, try to send them today because I am going to start working on that portion tonight. The plan is to get you guys the take home on Wednesday, and then of course the in-class portion is on Friday. And then next week the goal is to be working in here on blogs related to some topic in virology, and honestly I need to finish writing up the instructions for this, so I will get that to you as soon as possible. I'd like actually to probably try and finish at least most of it by Wednesday so that we can have maybe 5-10 minutes to talk about it, so that Monday when you come in here you're not spending your time trying to decide on a topic, you're actually getting working on it. So we'll talk about that later. That's the last piece though, and for that one I think that what I have planned is Monday, Wednesday, Friday are all work days. There's a day built in there for you to peer review another group's blog. I don't know if I'm going to do that or not, to be honest. I kind of want to, but I want to make sure that everybody has enough time to at least put something good together beforehand. And then to grade these, I am going to be recruiting some of my past students who are in grad school, med school, etc. to grade these ones. So, just so you know who your audience is on this. Me and others. Made in my mold. Alright. I think that's all I've got right now. Any questions before we get started? Alright. The stress level a little bit lower from the weekend? Yeah? No? Okay. For some, yes. For some, no. Great. Alright. Well, if anybody wants to come over to my place and help me grout the backsplash in the kitchen, you're more than welcome to. That can take out a longer. So, all right. So I just wanted to start out with this image here, just to remind you of the steps of the life cycle. We're not going to talk about this per se right now. I actually just want to kind of skip into this next one, where we're now looking at potential retroviral therapies. And one of the key points here is that pretty much every single step of the viral life cycle here is a target for retroviral therapy. And this is in some ways true for many different viruses. I've argued, at least in my mind, I think the reason why we have more retroviral drugs for HIV than others is probably twofold. One being that we've probably put more time investigating HIV than any other virus as a scientific community. That's my personal thought. My other personal thought here is that we don't have a vaccine, and so we need retroviral drugs to block the spread of infection. For some diseases like smallpox, polio, we've got incredibly good vaccines, and the rate of illness for those is incredibly low. Smallpox is completely eradicated. Polio, we're down to 3 countries that remain that have natural infections. And so for those reasons, I think that's why so much time and money has been invested into these retroviral therapies. Because many of HIV's proteins are unique, any of them could be potential targets that have relatively low side effects. There are some that are more toxic than others. But this here just shows some examples. inhibitors of the protease, which would block which step? When is the protease required? Maturation, exactly. So this wouldn't necessarily block the initial infection, but would block the ability of the virus to spread from one cell to another. If we wanted to use a therapy that would block establishment of the infection, what kind of targets might we want to, or what kind of enzymes might we want to target? That would be a good one. So blocking interaction maybe between CD4 and the virus, so blocking the attachment and entry. Could also include CCR5 inhibitors. Cellzentry is the main one that's on the market that functions in very much the same way. What other targets? That's the big one. Reverse transcriptase is the one that we have more drugs for than any others. The reverse transcriptase drugs can be broken into two categories. Ones that are known as nucleotide analog or nucleoside analog reverse transcriptase inhibitors, sometimes referred to as NRTIs. And then there are NNRTIs, non-nucleoside reverse transcriptase inhibitors. So some look like AGs, Ts, and Cs. Ribavirin would be a great example of a nucleotide analog. And then there are others that just inhibit by binding non-competitively to reverse transcriptase. Recently, there has been some investigation into using, or to identifying drugs that would block the RNase H activity of reverse transcriptase, so to prevent it from cleaving the RNA-DNA hybrids, which would block reverse transcription as well. Though those have not made it to clinical trials as far as I'm aware. What other steps might we want to particularly block to prevent establishment? Can think of maybe one other one. Yeah, probably integrase inhibitors would be really good as well. Within the past couple of years in the United States, there was individuals who have HIV-positive partners have been given prophylactic treatments of anti-HIV medication. The term that's used is PrEP, pre-exposure prophylactic. The main drug that's been used for this is known as Truvada, which is a combination of two drugs, both being reverse transcriptase inhibitors. So preventing that actual establishment of the viral infection. PrEP has also been used, or Truvada has also been used in cases where if you think you've been exposed to HIV, they suggest taking it within the first 72 hours can block the establishment. Much longer than that, and it's not going to have its effect in blocking the infection from occurring at all. So again, most of these targets are going to be ones that block entry, reverse transcription. Again, two categories here. There's at least one integrase inhibitor, at least one protease inhibitor as well. And those are kind of our major categories. Right? But again, there are other potential ones. Largely what researchers have focused on is trying to target those that seem to be most critical to the life cycle. And there have also been thoughts in trying to improve the antiviral response or the host immune system as well, boosting the immune system to block it. I just wanted to point out this graph right here, or this figure right here, to point out that HIV, like some of the other viruses we've talked about, undergoes mutation rather frequently. In the case of HIV, it's going to be during the reverse transcription step, when that low fidelity reverse transcriptase is being used. The idea behind retroviral therapy is to treat with 3 or 4 or 5 different drugs at one time, targeting different aspects of reverse transcription, integration, maybe the attachment and entry, And the goal here is to prevent any virus that has mutations to all of those different drugs, or mutations that render them resistant to any of those drugs from being able to grow and proliferate. In order for this to occur, the individual has to take the medication consistently throughout the remainder of their life. And as I said at the beginning of these lectures, we're now finding that individuals who are HIV positive are living just as long as the general public. So these retroviral therapies do work. The downside is, well, there's a couple of downsides. One, the cost is incredibly large. So it's cost prohibitive in some countries like Africa and others outside of the U.S. There's also price gouging, I will fully admit it. So the price of some of these drugs in the U.S. is about 5, 10, 20 times higher than it is in other countries because health insurance companies will pay for it. But that also can put a lot of money or a lot of burden onto the patient as well. And so this is just showing then, this graph is really showing a couple of things here. One, the mutation of the virus over time. Secondly, the amount of HIV viral particles that would be detected in the first few weeks and then over time in an individual that is HIV positive. And this correlating then with a drop in T cell number as well. And again, when we clinically define AIDS, it's based on a low T helper cell count and a high concentration of HIV within the individual or within the blood plasma. And I think, yeah, that's it. So that's the last slide for the semester. 2 weeks to go, and we're already done with slides. So what I did is I put in front of everybody this other sheet. I put together a few questions, and actually the first one is an older, I'd like to start with this first one. It's an old critical thinking problem that I used before. So I'll read it out real quick, and then what I'll have you guys do is let's start by working probably for 5, 10-ish minutes in groups or alone, and then we'll spend a little bit of time talking about it. So it says you were working, I think I was supposed to say you were working in a lab that focuses on HIV. Recently, a prankster came to your lab, removed all the labels from the antiviral drugs in the lab that you use. The boss, disheartened by the prankster, asked for your guidance and your infinite wisdom, which you have gained from this class. Remind your boss of that. You suggest that you should test the drugs for the effect on HIV's infection of macrophages. You will test one of these drugs to prove to your boss how indispensable you are. Drugs you have around are protease inhibitors, nucleotide analogs, fusion inhibitors, and integrase inhibitors. You plan to perform a Western blot on the left, PCR and gel electrophoresis on the right to detect protein and the provirus respectively. Luckily, you have anti-capsid or anti-CA antibody to use for the Western blot, and primers that bind to the LTR and GAG region of the genome can be used for a PCR to produce a 1.5 kb product. So based on the results, what kind of inhibitor is it, and justify? And then what other tests might you use to confirm that the drug does not affect the production of genomic RNA? Okay, so 2 parts here. All right, I'll give you guys, we'll start with about 5 minutes or so, see how that goes. And we'll chat. So 4 potential inhibitors here. Let's see, so we've got a chance for a protease inhibitor. Got a chance for a nucleotide analog, so I'll just refer to this as an RTI for the sake of shorthand. We've got fusion inhibitor, and then we've got an integrase inhibitor. What is the target of a fusion inhibitor? I don't think we've talked about this one. It wouldn't be tetherin. It's a good guess, but it wouldn't be. What protein is required for the actual fusion event? GP41. So that's the actual target of this one. There's only one fusion inhibitor. It's known as Fusion. Anyway. Okay, so where shall we start?
It's not fusion.
It's not fusion? Mm-hmm. Why do you say that, Grace?
Because it doesn't target GP41.
How do you know that, though? I'm not going to disagree with you, but that doesn't really... what I want you to do is, in this one in particular, start to use the evidence to argue for that. So what evidence do you have that says it's not targeting GP41? Well, that's probably more like out of design than anything else.
Because you're seeing capsid protein in these things, or you're seeing your product.
Which product?
Well, the anti-capsid antibody is recognizing that there's a mature virus, so it means it's entered.
Okay, so let's go along that line real quick. What other evidence suggests that entry has taken place? What's the other piece of evidence that we have? The DNA. And how has that been performed? It's just PCR. What does that tell us? What can we PCR and what can we not PCR?
PCR DNA.
And what can we not PCR? So what does that tell us about the virus? It's undergone reverse transcription, and what else has happened? It's probably integrated as well. So based on what we see to this point, it would at least argue that the virus has been able to make it through integration. That would argue against it being a fusion inhibitor. What else would it argue against? Anything else? Actually would argue against the the nucleoside reverse transcriptase inhibitor as well. So one way you could come to this is saying that, look, we know that it makes it to integration, therefore all of these upstream steps it's able to get past. Okay? That doesn't argue in favor of a protease, though. That just argues against the others. Maybe the trickster threw in an extra one that you don't know what it inhibits. So how do we know it's a protease inhibitor? Less cleavage. Go ahead, Sabrina. The Western blot. So what's true? Let's walk through the steps of how is capsid produced. So it's produced from GAG, and that comes again from the full-length genomic RNA, right? What has to happen to the full-length genomic RNA? After it's been translated, sorry. What has to happen to the translated product? Has to get cleaved by a protease in this case. So if we look at this blot, without the drug, we've got a 22 kilodalton product, which is we can assume is the capsid protein, because that's what the antibody is binding. Why do we have a larger size product with the drug? Not cleaved, and capsid is still part of that polyprotein, right? But it's also got with it nucleocapsid and matrix protein as well. So the argument here in favor is the Western blot, absolutely. With this particular inhibitor, the virus goes through all of the steps, even exits, but doesn't mature, and therefore, if we were trying to infect with this viral particle, it would be non-infectious. Okay. Questions on that process? So part of this one is just knowing the basics of the life cycle. And again, if I were to give this one to you on the take-home exam, you'd obviously have your notes available to you. And I would say look at all the notes, right? So the one hint I gave to Amelia is take a look at capsid. right? That's a key part of this. Most of what you need to search for is going to be embedded in these problems, okay? And then you can use deductive or whatever the other one is, the form of thinking in order to make your decision. So rule out or rule in. My non-mathematical way of saying it. All right. As a side note, what other test could you use to determine it doesn't have an effect on the genomic RNA? Or that you're not blocking any function or activity of the genomic RNA? This is more of a technique than anything else. Go ahead, Jonah.
Look for RNA for tubulin.
Okay. And compared to the genomic RNA?
Maybe.
Compared to the HIV genomic RNA? So tubulin would be a lovely control, right? I guess by gRNA I mean the HIV's genomic RNA, which is that full-length species, right? So one could argue, like let's say in figure, no, we won't worry about that. I guess the question that I'm trying to get at here is, how do we know the drug doesn't inhibit something about the transcription process? So how might we measure that? qPCR, reverse transcription in qPCR would probably be the most robust of the methods. You could also do just RT-PCR if you wanted to. It wouldn't be as quantitative, obviously. But yeah, reverse transcription with qPCR would probably be the best method. And then compared to something that shouldn't change, like tubulin. Good. All right. Let's do the last one. So for each of the drugs, predict which stage of the life cycle is going to be blocked. So I guess we don't have to do A because we've really already answered that one already. But do the other three. Valtigravir, AZT, and a REV inhibitor, which does not exist, which is why I just said REV inhibitor instead of an actual drug name. So, what step of the viral life cycle is going to be blocked for each of these? We'll go for another 5 minutes and we'll talk again. I think this would be a good one for multiple choice. Yes, Sabrina, it would be. Because...
Is this my extra set of questions?
No. No, but as I was sitting here, I think this would be a good multiple choice one, because I would expect you guys could probably figure this out with notes. This would not be a very hard one with notes to do. Really, the question isn't so much about the inhibitor, or you can look at this question, I guess, in two ways. One is, what's the effect of the inhibitor? The other way of looking at it is, what step is that protein actually involved in? Right? And whichever step is involved in is the one that's going to be blocked by the inhibitor. So, for example, with A, we've talked about how the protease is required for maturation. It's going to block the maturation process. How about B? Go ahead, Sabrina.
This is a random question, but
Yeah.
so if the virus is already exited and it's not able to mature, is that just degraded?
It would be inactive, yeah. And so when we say it's inactive, because GP120 and 41 aren't cleaved from each other at that point, and because all the proteins inside aren't cleaved from each other either, what we would find is reduction in the efficiency of attachment. And then if we do get entry, we're going to see a huge reduction in the reverse transcription in the next round of the life cycle. So it's really not doing so much for that cell that was infected, it's doing more to prevent the spread to other cells. And we might assume that with decreased attachment or decreased efficiency, it increases the likelihood that a macrophage or something else is going to come and phagocytize it to kill it or fully degrade it. So how about B there, raltegravir? Integration, biosynthesis, both would be fine answers. AZT?
Reverse transcriptions, biosynthesis.
Yeah, I would say that works too. Reverse transcription, the biosynthesis. Sabrina, go ahead.
That mechanisms are that just that causing more like base pair mutations?
That's a good question. So some work in that way to create more base pair mutations. Some of them are a little bit different. They're what we call chain terminators. And I'm glad you brought this up because this was something I actually wanted to address and forgot to write down in my notes. So let me try to draw this mechanism out real quick. And you guys have seen time and time again, I'm sure, structure of nucleotides. So here's our phosphate. ROH, if it's DNA, there's an H, there's a couple H's there. And then we have our nucleotide base, which I'll just refer to as R right now. So some of these drugs are what we call chain terminators. In other words, they have the 5' phosphate, but they lack the OH group on the 3' hydroxyl, or the 3' carbon. Lacking that hydroxyl group would prevent any additional dehydration reactions from occurring, and thus would prevent the extension of that DNA. So that's how some of these at least work, Sabrina. So whereas ribavirin is increasing mutation frequency, some of these drugs would be just blocking the addition of nucleotides and kind of lead to like an abortive reverse transcription step.
So like that nucleotide would fit into like the active site of
Yes.
and then be integrated and stop the
Exactly, and block any further nucleotides from being added to it. Exactly. Now, does Francie talk about sequencing at all in genetics? Sanger sequencing?
I kind of want that out.
Oh, gosh. So Sanger sequencing, not what we did, but Sanger sequencing is where Sanger sequencing is basically the way that it was done before, and it gives you good 800 base pair reads or so, but they use chain terminators in Sanger sequencing. So you have them at really low concentrations so that they're not blocking the replication all the time, but that you're getting essentially different length DNAs. And this would look like if you were to line them all up. Well, those chain terminators also have a fluorescent marker on them. And so that as they're running off of like an HPLC or some other column, you can detect the fluorescent marker, and that tells you, is it an A, is it a G, is it a C, or is it a T? So, okay. Oh yeah, I would have done that if this was a molecular biology course. We would have gone into the method more. But, you know, it's virology, so. Grace.
So this nucleoside analog is integrated in like the middle of the genome?
It would be integrated somewhere during the process of the reverse transcription. And the where it is going to be completely random. But the goal, again, with those reverse transcription inhibitors is to either block the reverse transcription from being completed fully, or to create mutations to a point where it's like that catastrophic error that we talked about with Ebola.
Wouldn't that be harder in HIV because it is so
error prone?
Yeah.
You might argue that it would be. Yeah. I mean, HIV seems to be more willing to undergo some mutations, whereas Ebola seems very fixed, right? That was one of the takeaways from that paper that we read. Sabrina.
So do the non-nucleoside inhibitors just inhibit enzyme function as a whole?
They do. So they're non-competitive inhibitors. I mean, you might see the term allosteric inhibitor as another way. So they're going to distort the active site so that those DNA or deoxynucleotide triphosphates are unable to make it to the active site. And you could argue that those might be a little bit better because then you don't have necessarily a competition for that active site. And blocks probably all of reverse transcription instead of, partially through or something like that. How would an inhibitor of Rev function? Maybe?
Isn't Rev responsible for taking the larger chunks in and out of the nucleus?
That's true. It's before maturation, though. Biosynthesis, maybe assembly, maybe even pre-assembly, because it's really required for, as you're pointing out, it's required for the export of those partially spliced or those unspliced RNAs. So it's required to export the genomic RNA. It's required to export the forms of the RNA that are going to produce GAG and POL. You could say that this is only biosynthesis, and specifically repression of Rev or inhibitor of Rev is going to prevent assembly, exit, and maturation from occurring altogether. It would still allow for some viral proteins like VIF, NEF, VPR, VPU to be produced, and Rev and TAT also, I suppose. But it would block the translation of the GAG and the POL precursors. So ideally, you wouldn't get out any infectious viral particles.
And you said this never has been explored before?
Not as far as I can tell, which is why I just came up with a no name for it. Zactigravir or something like that. Pratigravir.
Why do you think they haven't?
That's a tough question to answer sometimes. My guess is that maybe they've looked, but that either they can't find inhibitor. There's not been an inhibitor that's designed that actually can work so far. And such an inhibitor would have to be able to not only make it into the cell, but also would have to be able to make it into the nucleus as well. And I don't know if that adds an extra layer of complexity to the question, to the issue. I mean, somebody put on a drug like that would still probably experience some of the, depending on how many of their cells have provirus, would still probably experience some of the effects of immunocompromisation, and that NEF and I can't remember if it's VPR or VPU, both block CD4 expression. So you might still experience some of the immunocompromised effects of HIV based on that. It obviously wouldn't be as severe as killing those cells, but... Any questions? I want to just see if this has brought up any questions for anybody as we're going through this process. I suppose one way to summarize the antiviral drugs, it's some ways intuitive what the effect is going to be, and that's really all we're focused on right now. We're not talking necessarily about how they are functioning or where they're binding. We're just focused on what's the effect. Okay. And that is a rather intuitive thinking process if you understand at what point of the life cycle are these different proteins involved in. So that would be one thing that I would say between now and Friday you want to make sure to have an understanding of. There's still a lot of investigation into the retroviral drugs. Obviously, this is kind of one of those things where it's a lot of money. I mean, you could argue why companies spend time and money on this. If somebody has HIV, how long are they taking these medications? The rest of their life, which is the same reason why I argue why people put so much money into diabetes medication, heart medication, etc. These are lifetime chronic drugs to be used. And this has been a big issue in the U.S. and outside as well. So, again, Bush the second created a program called PEPFAR, which was designed to bring antiretroviral drugs to sub-Saharan Africa, the area that's been most greatly hit by HIV. And accessibility to those drugs has differed based on the country. Some countries have done very, very well to reduce rates of infections. Others have not done so well. South Africa is one where actually, as the rates in the rest of sub-Saharan Africa were decreasing, South Africa's rates were just climbing. And this came from not so much cultural, but really from a political perspective. The leader of the country at the time was an HIV denier. In other words, he did not believe the arguments from where HIV came from. He thought it was a product of Western medicine. And so because of that, South Africa didn't take many of the same strategies as other countries did to reduce rates of spread. There's some very, very... I should point out one other area where these antiretroviral drugs are very important: pregnant mothers. So if you think about the birth of a child as they are passing through the birth canal, we've talked about how the spread of HIV is through direct contact, typically blood from one person to another. Well, if there are microabrasions, a child can pick up HIV as they are passing. Also, they can pick it up through, we know that mother's blood is used to help provide nutrients, etc., to the fetus. HIV can spread through that way as well. It's been found that if mothers are put on retroviral drugs, it reduces the chances for HIV to pass from mother to child down to about 5%. There are arguments that if we have a vaccine as well, it might reduce it down to about 1%. But those are just, right now, hypothetical numbers. And this is an issue that we still see in our country, too. So I don't know if anybody recalls, but about 2 years ago there was an outbreak in Indiana. And the population of individuals where this outbreak occurred, it was kind of a perfect storm of bad conditions. Unemployment was high. There was a drug epidemic in this area of Indiana. It was a relatively rural area that didn't have a lot of, I suppose, maybe the way to put it is that it didn't have a lot of physicians in the area to keep track of individuals who were HIV positive, and those who were using drugs were sharing needles. A lot of cities in the United States have implemented needle exchange programs. The thought is that by introducing these needle exchange plans, it'll at least reduce the rate of HIV. The goal in those plans is not to reduce the rate of drug use. That comes separately, but just to reduce the spread of HIV and hepatitis C and other bloodborne pathogens. And in Wisconsin, I do not think that it is a statewide, from the government level plan, but there are different programs in the state, like what was known as the AIDS Network is now AIDS Resource Center of Wisconsin or something like that, that do run these needle exchange programs as well and do regular pickups. It's completely anonymous as well. So there's a lot of issues to look at with HIV just beyond what we know about with the virus as well, and thinking about healthcare, and again, a lot of different voices take play in this conversation as well. Any other questions before we head out for the day? All right. So give me those questions if you have them for extra credit, and then we will on Wednesday come with questions. If you've got them, come with questions. I have sheets from the posters, so I just got one for each group. I've put on notes here that are not only my own, but then also from the faculty that were helping me grade these as well. So you don't know whose comments are whose per se, but they're on there. All right. Give that to you. Grace, here's yours.
Did you see our great farm poster?
Bye, Bill. Hey, you.